So we are going to start our topic direct evidences of DNA as genetic material. So moving with the topic, first of all, what are the direct evidences? DNA is genetic material as it was clarified by the year 1953 with various experiments. For clarifying this, there were many direct and indirect evidences. Some of the direct evidences were like bacterial transformation, bacterial infection by bacteriophage, as well as some others were there. So moving with the first one, I am going to start with bacterial transformation, which I will prove with Griffith experiment as well as Avery, MacLeod and McCarthy experiment. The bacterial transformation. First direct evidence for DNA as genetic material is from Griffith experiment that was actually performed in the year 1928. In this experiment, it was observed that there are two strains of bacterium Staphylococcus pneumoniae. The first strain is R cell and the second strain is S cell. The R cell produces rough colony, S cell produces smooth colony due to presence of a gene that produces the polysaccharide to make the surface smooth. But this gene is either mutant or inactive in R cell. It was very well known fact at that time that R cell could not change to S cell on its own, but the S cell could easily change to R cell just by mutation in that gene. When Griffith was performing the experiment, and another important observation that was known at that time was R cells were avirulent and does not cause any pneumonia in the organism, in the host organism, but the S cells are virulent that can cause pneumonia. Hence, when Griffith introduced R cells into the mice, it does not causes any pneumonia in the mice, and from that mice, when the extract was taken out, only the R cells were observed. But when S cells was injected into the mice, it got symptoms of pneumonia and it got died. And when extract from this died mice was taken out, it also shows that S cells were present. When heat killed S cells were injected into the mice, it produces no symptoms, mice remains healthy, and only the living mice extract was obtained and no strain cells either R or S were obtained from the mice. But an important observation was made when living R cells and heat killed S cells together were injected into the mice. The mice shows the symptoms of pneumonia, it got died, and when extract was taken out from the mice, it shows both R cells and S cells in the extract. But we have not injected living S cells. How could these new S cells were observed? R cells themselves cannot mutate into S cells. Then what was the reason that these new S cells were observed in the extract? From this, the hypothesis was made that there was something that from heat killed S cells moved to the R cells to convert them into S cells. This is something was what was proven to be DNA by Avery, MacLeod and McCarthy experiment. So without wasting any time, we are moving to our next experiment, that is Avery, MacLeod and McCarthy experiment. In this experiment, that heat killed S cells that were known to be causing the conversion of R cells into S cells was taken in a test tube, and from this lipid and sugar content was extracted out. Now, there were present three type of components in the mixture in the test tube. These were the proteins, RNA, and DNA. This test tube components were divided into three parts, three samples. In one of the sample, they added proteinase, which digested the protein. In the second sample, they added RNase, which digested the RNA. And in the third sample, they added DNase, which digested the DNA. Due to digestion of proteins, now the test tube is left only with RNA and DNA. When this sample was injected or grown in colonies, S cells appear. It shows it was grown in colonies with R cells. S cells appear. It shows that protein is not a transforming agent because protein was not present. Still, transformation was happening. In the second sample, when RNA was given, RNA was digested. Now the tube was left with protein and DNA. When it was injected or given to the R cells, the S cells still appear. It shows that RNA is also having no role in transformation. But when DNA was given, DNA was digested, protein and RNA was present. The R cells mixed with this mixture shows S cells do not appear. In spite of the presence of protein and RNA, S cells do not appear. It means protein and RNA are not having any role in transformation, but the DNA is having. Hence, DNA should be the genetic material for the organism. Then, second evidence for DNA as a genetic material was given by bacterial infection when bacteria was infected by bacteriophage. It was observed that in bacteriophage, phosphorus is present only in the DNA molecule and none in the protein part. Similarly, sulfur was present only in the protein part and none in the DNA. Then one sample of bacteriophage was labeled with radioactive phosphorus 32P and another sample of bacteriophage was labeled with radioactive sulfur 35S. These two bacteriophage samples were independently added to bacterium culture and then they immediately grinded up to detach the viruses. Then the sample was taken out in a test tube, centrifuged, and it was observed that in the first case where radioactive phosphorus was given, all the radioactivity was observed with the bacterial cells and none with the supernatant liquid. Similarly, in the second sample where radioactive sulfur was given that was present in the protein pool, all the radioactivity was observed in supernatant liquid and none with the bacterial cells. It shows that it is the DNA that enters the bacterial cell. Hence, in the first sample, radioactivity was observed with bacterial cells. The protein code does not enter the bacterial cell to transform it or to produce its copies. Hence, all the radioactivity was observed in the supernatant liquid. This definitely shows that DNA is the genetic material that carries all the information to produce its progeny. But none of the information was present in the protein code that to direct the synthesis of new progeny. Hence, these two direct evidences proved that DNA is the genetic material. Thank you.